Hi there. Welcome to Microbiology and Immunology. Can I check that you can hear me in the chat, please? Just let me know if you can hear me okay. Fabulous. Thanks, guys. Right. Okay, so new session. Apologies for the communication issues last time. We got a bit mixed up with when the sessions were on, so sorry about that mess up. And new unit. Let's go into slideshow. So what we're going to be looking at in this is obviously the immune system and microbiology, because quite often what the immune system is doing is responding to microbes. Okay, 1 second. I'm just getting somebody unable to hear. Let me just respond to that message. Okay, hopefully they'll sort that out. So we're going to have a look at the immune system. We're going to have a look in this session about some of the components that are circulating in the blood, because a lot of the immune system is circulating. Some of it is fixed. So we had a look in quite some detail a couple of units back at the liver, and those specialized cells, the Kupffer cells in the liver, didn't we? So some of it is fixed and some of it is in other organs as well. But a lot of the components to the immune system are circulating. We've talked about loads of them already. We keep talking about the macrophages and their ability to carry out phagocytosis. So what we're looking at today is the composition of blood, the function of the white blood cells. Not looking at platelets today, but we will look at platelets. We're going to have a look at phagocytosis again and the adaptive immune response this time. We've had a look at some parts of the immune response, but not the adaptive immune response. And we're going to have a look, if we get to it at the end, at the 4 different types of immunity that we have in our system. Okay, let me just check that message because I can see another message has just come in. Okay, good. Right. So first thing we need to know about our immune system is we have lots of layers of defense that have to be breached before we even get to activating our immune system. So all organisms have strategies to defend themselves. This one's really clear. This one's, this layer of armor that you can see on this animal is really to defend itself from predation, to be honest, but it would be very good defense against pathogens as well. So many of the dangers that we face are much, much smaller than that sort of armor is going to help. Things like bacteria, viruses, fungi, parasites, and some really horrible worms as well, which we'll have a look at later. So we have, not later today, later in the unit, we have developed a multi-level defense system. The immune system is extremely complex, and we're not going to go into massive levels of depths on all of it because it's a degree. You can do a degree in immunology. It's a huge, huge area of biology. But we're going to have a look at an introductory level. So the defense system, before we get to the actual immune level, consists of the skin and the mucous membranes, which we've already talked about. And then we get to the immune system. So the first line of defense are the barriers. They're non-specific because it's things like skin. And if you think about it, the main routes of entry into the body are cuts, so penetrating the skin. We can get wounds, we can get burns, we can get abrasions. So anything that cuts open the layers of the skin can allow entry into the body. We've got the respiratory tract, and we've talked about that in an earlier unit when we were talking about the mucous membranes, moving anything that gets trapped in the mucus into the back of our throat where we swallow it. We've got the digestive system. Again, another exchange surface where you've got those goblet cells that we talked about releasing hydrochloric acid and the proteases, but also things that we haven't mentioned yet as the reproductive organs. So you'll all be aware of sexually transmitted infections. It's a really common route of infection into the body because obviously you exchange bodily fluids with another person during intercourse. So obviously that's a route of infection in. And our eyes are quite a common route of infection, really common in children because they put their fingers in their eyes, or we all do, if you get an itchy eye, we always all rub our eyes. But with children, when they're less likely to be thinking hand hygiene, not washing their hands, things like conjunctivitis spreads really, really quickly amongst young children. So the eyes are a route of infection that we don't actually think about that much. We're all very familiar with the idea of washing a cut out if we get a cut so it doesn't get an infection. We're all very aware of respiratory tract infections, particularly in the winter when viruses are common, and digestive upset. We're drummed into us at school about sexually transmitted infections to make sure everyone is educated on that, but that one is probably a bit less considered. So we're going to have a look at the skin. It's an organ. It's our largest organ, and it's really important in terms of defense, of stopping entry of pathogens into the body. So 1 centimetre square contains loads and loads of skin cells, 10 hairs, muscles, 100 sweat glands, 15 oil glands, 200 nerve endings, including 25 pressure sensors, 12 heat sensors, and 2 cold sensors. So there's an awful lot of sensors going on in that organ. So part of the nervous system, sensory detection is going on there. Heat sensors, cold sensors, pressure sensors. Then we've got all these glands. So these are hormonal. So loads of glands. We've got hairs and we've got muscles. So there's an awful lot going on there. And you can see that by looking at this cross section. So hairs, sweat pores, you can see it going through that top layer of the skin there. And then if you look down here, we've got muscles. These erect the hair when we get cold and trap a layer of air to help keep us warm. Not particularly effective in humans, to be honest, but it helps a bit, but pretty effective in animals. Got oil glands to help look after our hair. Arteries and veins to supply the tissue with nutrients and oxygen. And then we've got a sort of shock absorbing layer at the bottom there, fat layer. So really quite complex. We're going to have a look in a little more detail now at these structures. So the 3 main layers that we're interested in, we've got the epidermis, the dermis, and the subcutaneous layer. So epidermis, dermis, subcutaneous. So that's basically mostly fat, that subcutaneous layer. But these two, the dermis and the epidermis, we're going to have a little look at now. So the skin is almost impenetrable. We're pretty safe, to be honest, from infection via the skin, unless we get a cut. We tend not to get anything burrowing through. It's pretty good, pretty safe. But obviously we do get cuts and scrapes and things fairly frequently. One of the ways the skin protects us is that it has quite an acidic pH. So somewhere between 4.7 and 5.7. So it's pretty acidic. 7, pH of 7 is neutral. So it's got quite an acidic pH. That's due to the oil that's secreted onto it and the sweat as well. So that's not a very good environment for bacteria to live on for a start, or fungi. It's not particularly hospitable as a living environment for pathogens. It really does inhibit microbial growth, this acidic pH. The sweat that we release through our sweat pores contains a lysozyme enzyme, which is an enzyme which breaks down bacterial cell walls. So that's also really helpful. That's just in our sweat. Also, our tears contains lysozyme as well. But we're talking about the skin, so let's talk about the skin. So the top layer then, the epidermis, is about 10 to 30 cells thick. There's a image here as well. You can have a look at. This is the epidermis towards the top here and the dermis towards the bottom. So this is a actual slide of a microscope slide here. So dermis at the bottom and the epidermis at the top. It's about 10 to 30 cells thick, which isn't very thick, to be honest. And then we've got this layer here, which is continually abraded. That's the top layer. Okay. You can see this is like a cross section. This square here is a zoomed in version of that down there. So the top layer of the epidermis, which is called the stratum corneum here, is continually abraded. That means it's continually scraped away. So it's just scraped away just by touching stuff like our clothes or just even the air. Anything we do with our hands will be wiping away skin cells. So you'll have probably been aware of this from watching like crime dramas on TV, that you can pick up DNA by swabbing it. You can pick up cells because if you touch something, you will be leaving some cells behind because your top layer of your skin is continually abraded. It's continually scraped away. So the dead cells that are continually scraped away on this top layer of your epidermis are replaced by cells from this lower level of your epidermis down here. It's the base layer, if you like, the stratum basal layer. These are continually dividing and replacing these top layers that get shedded as we go about our daily life. So these cells here, this bottom layer of the epidermis, remember it's this square up here that's zoomed in. So this bottom layer of the epidermis here is continually dividing, and they're the most actively dividing cells in our whole body. And that takes some doing because our red blood cells are replaced at a hugely fast rate as well. Because our red blood cells, remember, we learned about how they're adapted for their function. They have no nucleus, so we can fit more oxygen in, more hemoglobin to carry oxygen. So because they've got no nucleus, they can't repair themselves. So they're replaced really quickly, but these are replaced even quicker than those. So continually dividing to replace this continually shedding top layer of skin. So we've always got a good new layer of skin. And another thing about the skin is when the cells are produced, they produce something called keratin, which is a protein which toughens up the skin as well, makes it harder to damage and keeps it as a nice, tough, impenetrable layer. So cells remain at the surface. So, it's a fairly, what was, reasonable layer. They will remain there for about a month, each individual cell. Obviously, there's always cells being removed, but each individual cell will live there for about a month before it gets scraped off, assuming you don't damage it in any way. But in some conditions like psoriasis, for example, the rate of this production of new cells is really speeded up. So in psoriasis patients, it can be either every 3 to 4 days that you're getting replacements of these cells. So the shedding of the skin cells is increased. So that's the epidermis. The lower skin layers are there for support and for insulation. So the dermis is many, many times thicker than the epidermis. That's this layer here. It doesn't look many times thicker on this diagram, to be honest, but it is. It's a lot thicker. And this provides a lot of structural support. And you can see on the diagram here, there's loads more going on inside this layer, this tissue here. This is just skin cells, This has got nerves. You can see muscles. There's so much stuff going on in this this next layer down here. So it's nerve endings, blood vessels, muscles for erecting the hairs. And this is the bit that gets wrinkly as we age as well. And it's actually what we harvest, I suppose, when we get leather from animals. That's the layer that we're using. And then underneath that, we've got this layer here, the subcutaneous layer, and this is basically adipose cells, and that's fat. Adipose tissue is just fat. You can see it looks like, well, it looks like fat when you cut meat, doesn't it? And that's exactly what it is. It's a layer of fat. So this acts as a shock absorber. So if we get bumps and knocks, it helps protect anything underneath the layer of the skin. So it helps protect our muscles, helps protect our organs, it helps protect our bones, depending on where the skin is. There's various thickness within the body. Obviously, we tend to deposit fat in certain areas, like around the tummy, for example, around the abdomen. We're more likely to deposit fat there. But it also provides insulation. So that's really important as well, keeps us warm. So that's the skin, which was one of our external barriers. And another external barrier we've talked about quite a lot already over the course. We've talked about the mucous membranes. So what's important about the mucous membranes is they tend to be made of epithelial cells, and these have really tight junctions, which means they're really tight together. There's not many gaps between them that are easily penetrated, so it's not easy for anything to get through those gaps. In the lungs, we have air flowing over the surface, or in the stomach, those are the two mucous membranes we've talked about. The stomach has fluid moving over the surface, like gastric juice in the stomach, for example. The movement of the mucus in the respiratory tract, remember we have those cilia that move in a wave-like fashion that move the mucus over the mucous membranes inside your respiratory tract, and they help to move it towards the back of your throat to the glottis, where it can be swallowed. It then goes down into the stomach, where there is hydrochloric acid, another mucous membrane actually, so that needs to be protected from anything that you might have inhaled as well as anything you might have eaten. But we've got this really strong acidic environment in the stomach, which helps kill off anything that you might have swallowed, whether it's come from your mucous membrane secretions of your respiratory tract or you've just eaten it. It will be protected because of this nasty acid environment. But we've also got these antibacterial peptides, which we can find in the gut, and we've got protease enzymes in the gut, which break down proteins. So any living thing tends to have lots of proteins making it up. So all of those mucous membranes have also got adaptations, structural adaptations that protect them. So the skin, very acidic, loads of layers, difficult to penetrate. The mucous membranes, very tight junctions, loads of chemicals like hydrochloric acid, proteases to protect them, continually moving mucus over to trap any pathogens. Then we've got other mucous membranes, for example, vaginal mucous membranes. There are lactobacilli which live there and should live there. You know how we have gut bacteria that should be there. Also, the vaginal tract should have bacteria there. They produce these lactobacilli, produce lactic acid, which keeps an acidic environment in the female reproductive tract, which helps protect it from bacterial infection as well. So the main external surfaces then, other than the skin, that we need to think about is digestive tract, which has got saliva, which has lysozyme in it, which breaks down cell walls. The hydrochloric acid in the stomach, really nasty to live in. The respiratory tract, it's got this mucous membrane, so we're moving that mucus over the surface, trapping bacteria, cilia sweeping it over, and then it gets swallowed, where it goes straight into the stomach. Obviously, horrible environment because of that acid, which will kill a lot of stuff. And then the vagina, naturally low pH acidic mucous membranes to help prevent bacterial growth there, and the lactobacilli bacteria, which should naturally be there to help kill off anything. So that's our barriers. And when our barriers get breached, so for example, we get a cut, or unfortunately we've breathed in some pathogens or eaten some food poisoning pathogens, we have a non-specific immune response. So this is the response that we've talked about quite a bit before. We said it's non-specific. We don't know what we've eaten. We don't know what we've breathed in. So we're just going to hit it with whatever we've got. It's not specific. It's not right. I know it's that bacteria. So I'm going to hit it with my immune system, which is specific to that bacterium. It's just, I haven't got a clue what I've breathed in. It might be pollen, it might be bacteria, it might be a virus. Just get it out of there. So this is non-specific. These are all different types of white blood cell, which are involved in all different types of immune response. They're produced in the bone marrow. They circulate in the blood. They circulate in the lymph. Remember, that's the blood that drains into the tissues, but not the white blood cells. Loads of different types. Neutrophils we're going to talk about. There's monocytes, lymphocytes we're going to talk about. Basophils, so many different types, but they've all got pretty much similar jobs. They're all involved in helping us if we get something into our system. So this part of the immune system is mobile and circulating. So it can move and it circulates around our circulatory system. There are some fixed structures, though. For example, there are the lymph vessels are part of our immune system. The lymph nodes we talked about in an earlier unit, these are a site of antigen presentation. So these are somewhere where the antigen presenting cells will present the antigens to immune cells. I'll just talk you through that again in a minute. We've got the spleen, which is like a very big lymph node. So again, somewhere where the immune system does its antigen presenting, so gets the immune response going. We talked about the liver, which can filter the blood directly from the gastrointestinal tract. Remember that hepatic portal vein, all of those blood vessels that are taking in our nutrients in the gut, they go straight to the liver. That's the first place it goes to. So the liver's got a really important role in filtering the blood. And again, that's a site of antigen presentation. So let me just remind you of what's going on there. Just got to move a little box so I can see what I'm talking about. So we have done this before, but just remind you. So what we've got here is a white blood cell, and it's covered itself in antigens that it's got from an invading pathogen, and it's got this special molecule here called the major histocompatibility complex, and it will take an antigen and show it to this immune cell, and this immune cell can then go on and generate an immune response to help get rid of these pathogens once it's noticed that they're there. So that's one part, antigen presentation of the immune response, but there's an awful lot of things going on. So the non-specific defenses consist of, once the defenses have been breached, so your skin's been cut, something's got in, you've breathed something in, you've ate something. It's any invader, we don't know what it is yet. They can ingest cells by phagocytosis, which we know about, but we're going to have a look at. We can produce antimicrobial proteins that kill pathogens. We're going to have a look at that. We can raise an inflammatory response, so inflammation, and we can have a temperature response. So all of this is stuff that we can do, 4 things that we can do when we get invaded by something and we don't know what it is yet. So phagocytose it, kill it with antimicrobial proteins. We can raise the inflammation response and raise the temperature response. So these non-specific defenses, the cells that can destroy the invading microbes, involve these guys here. So we've got a macrophage, we've got a neutrophil, and we've got something called a natural killer cell. All types of white blood cells, and they all look pretty similar, to be honest. It's only really their role that can kind of tell them apart, is what they're doing. So we've got the macrophages, the neutrophils, and the killer cells. So once that first line of defense is breached, we've got these cells circulating and they will kill the foreign cells. So the surface of the cells, our cells, has got self markers. We've talked about these before. They identify them as part of our body. They are extremely variable in the population. And sometimes bacteria might mimic some of these cells, but they can't mimic everyone's cells because there's variability in the population. You might get the odd bacteria that gets through because it's doing a pretty good job of mimicking some of these structures on the surface that some people have, but not all people will have that same cell. So when we had a look at this before, we had a look at these antigen presenting cells. And remember what they do is they display bits of the antigen, fragments of the antigen on their major histopathology complex molecules, and then they present them to the T lymphocytes, and that will initiate an immune response. Okay, so here's our pathogen or virus. Here's our macrophage. It'll break up that virus into little bits, and it will put these antigens on its surface and show them to this T lymphocyte. And this T lymphocyte will then activate some other cells. So either a killer cell, which we're going to have a look at, or B lymphocyte, which we'll have a look at later on as well. So this is the first kind of thing that triggers this major histopathology complex presenting, which we saw over here, look, presenting these antigens that it's got from breaking down pathogen, sticks them on its surface to a molecule called the major histopathology complex, sticks the antigens on its surface and shows them to this immune cell, and then the response gets triggered. So the macrophages, the neutrophils, and the natural killer cells are the ones that will identify non-self cells because they don't have that marker that identifies them as self, which inhibits an attack. If we fail to identify our own cells as self, and our immune system starts to think they're foreign, as in not part of our body, it will start attacking them, or it could start to attack them. And that's how autoimmune diseases develop. So of our non-specific defenses, these 3 cell types that are help us with our non-specific defense, we have the macrophage or macrophage. Some people say macrophage just literally means big eater, macro, big, phage, eater. And as we know, we've seen this image quite a few times now, it kills microbes by ingesting them in this process called phagocytosis. So it loops its pseudopods, that means like false legs, around the pathogen, and it ends up in a membrane-bound vacuole, which is filled with lysozyme. Remember, that breaks down the bacteria wall. This will basically digest the bacteria down. It will also break down anything else that it happens to engulf, like cell debris or dust or pollen, whatever. So these macrophages are just going around in your blood, waiting to come across something that shouldn't be there. Then we also have neutrophils, and these ingest most prominently bacteria by phagocytosis. So these are able to undergo phagocytosis as well. And they also release chemicals from their granules. That's what those little dots are. They're just granules. They just stain up differently when you look at them under the microscope. So that's how we can sort of tell they're different, because they look a bit sort of spotty, granular, we'd say. Just got a little box in my way a minute. 1 second. So another thing that the neutrophils do, they release chemicals from their granules, and these are called reactive oxygen species, or they're sometimes called ROS for short. These are not very pleasant chemicals. For example, hydrogen peroxide, that's one reactive oxygen species. And what they do is they cause something called an oxidative burst, and that directly kills and damages the pathogens. And it's these reactive oxygen species. You know, when people say, oh, you need to have a diet that's rich in antioxidants, like green tea and blueberries, and they're all things that are rich in antioxidants. Those are things that soak up reactive oxygen species because they're not very good for our body either. But reactive oxygen species like hydrogen peroxide can directly kill and damage pathogens, and they will kill any bacteria that are even in the vicinity of that neutrophil. They're pretty nasty. Then the last type of cell that's involved in this killing, non-specific killing of microbes are the natural killer cells. So what they do is they don't directly kill the microbes, but they kill off the infected cells. These are particularly good for virus infected cells. What they do is they create a hole in the membrane of the infected cell. Water rushes in, cell bursts, and then it's game over for that particular cell. They're also really good at getting rid of cancer cells if they find them as well. So that's your three types of cells that are involved in the non-specific response: macrophages and natural killer cells and neutrophils there. Then the second thing that the second non-specific response that goes on is there's some special antimicrobial proteins that kill pathogens that are just floating around in our blood. So this looks quite complicated because it is, quite frankly. But these proteins, there's loads of them. There's about 30 altogether, I think. And they just float around in our blood. They're called the complement system. They're just basically a way of triggering the immune system. They complement or help the work of the antibodies and the white blood cells that are in our blood. I'm going to show you a little video in a minute which kind of helps to explain it. But basically, we've got all of these proteins, they're all floating around in our blood. You get a cascade happening where one protein gets churned into another protein, gets added onto another protein, gets added onto another protein, and eventually you get this sort of hole forming inside cell, which will burst it. So that's the complement system, special proteins that work together in this cascade of protein building to kill off cells. So the complement system has to get activated. You get a big chain reaction. We're going to have a look at it happening in a video now. And that helps to destroy pathogens. So yeah, it destroys them by making a hole in the foreign cell, helps it die by getting water to fill in, swell in. And it will also start the inflammatory response as well, which is another response that we were talking about a minute ago. It will stimulate histamine release, and that increases the permeability of the blood vessels, and that can help white blood cells move in and out of the blood vessels, and it can attract phagocytes to an area as well, because they give off a signal that will move more immune cells in. So we're going to have a look at this now. Now, I don't think you can hear the sound, so apologies for the ads coming on. I'll skip them as soon as I get a chance. Right. Can I just check? Can you actually hear the sound of the video? Can you let me know in the chat? No. Okay, that's fine. As long as I know, I can tell you what's going on. Right, sorry about that. I'm just closing some boxes so I can see the screen. Okay, so those are our cells. That's our skin on the left. Well, there you go, immune cells on the right. So these are the proteins. These are the proteins in our complement system that are just floating around in our blood. And you can see by looking at the screen in a moment when I press play again, there's loads of them, loads of different proteins, loads of different shapes. So it's a really complex way, and we're just sort of looking at a summary of it. So there they are, all floating around in the blood. Loads of them. So there's a bacterium. So all of these other things that we know about antibodies and that they just activate our complement system. And essentially what that slide there was saying is that once they can just get triggered to go off at any point. And when they do, it's like tiny bombs going off everywhere, sort of going, right, activate, 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 essentially. This is just the start of the video, the music. So complement system does three things. It will slow down the bacteria, it activates your immune system, and then it rips these big holes into the bacteria that causes the water to rush in so that they die. So they're just proteins. They're all floating around. Loads of different structures, loads of different shapes. So when they get activated, they change their shape and that helps them to bind. So you can see that one binds now because of its shape. So that one's doing nothing, and then it gets activated, changing its shape, and then it can activate another protein, which can activate another protein, which can activate another protein, which can activate another protein. So we've got a cascade going on of proteins being activated one after another. It's like when you light a match in a box, that quickly ignites the surrounding matches. So you get this knock-on effect. It's really complicated in reality. So obviously we're just looking at quite a simplified version of it. So you get a cut, load of bacteria gets in via your skin barrier. That's your first complement protein. It's called C3. It's like the first match in that example we were given. So that's been activated now. So it can happen randomly or it can get triggered to happen. So this one's called C3. It breaks into two parts. You don't need to know these names at all. One of those parts is going to go and stick to bacteria, fungi, and viruses. If it doesn't find a bacterium quickly, it just dies off. It's a protein, so it can't die, but it doesn't work anymore. When the protein binds to a pathogen, it changes its shape. It lets other proteins start to bind. So we've got a cascade now going on. Loads of things are happening. Loads of proteins are binding. And now we've got a protein on the surface of our pathogen, which is activating loads and loads of complement proteins. So that is now covered with complement proteins because all of the complement proteins are getting attracted to it, which they're saying it's like getting swarmed by a load of flies. So remember the part that broke off, that goes off into the bloodstream, and that's going away from the site of activation now. It's flooding away off into the bloodstream. But what that's going to do is it's going to leave a trail back to that bacteria for other immune cells. So they can realize where that complement protein has come from. They'll follow the trail back so they know where the infection is. So they'll get to the cut in this example. So the first to arrive are the phagocytes. So they'll start engulfing, as we know. Then they'll break it down with the lysozyme once they've engulfed it. But they're actually quite hard to catch hold of. So the complement system helps them be engulfed as well. So these complement proteins that have stuck to the bacterium actually help the phagocyte grab it as well. So they're absolutely covered like a swarm of flies, but they're saying it's actually a little bit more like being covered like a swarm of wasps, because what these complement proteins can do on the surface is they attract other complement proteins which start to push through the membrane and they make a hole. That causes all the water to surge in and it just sort of falls apart really because it's full of water. Lysis, it's called. So that's how the complement system works. So that saves us from infection. It's actually really useful against viruses, more useful than bacteria. And the reason for that is because viruses can only inside cells, so they have to go from cell to cell. And if they can get stopped while they're going from cell to cell, and that's what the complement system does, it will stop the infection of new cells with the virus. So essentially, what this bit is saying is the reason we actually get sick, because you'd think that was such an effective system, like we really shouldn't be getting sick. It works really well. Is because bacteria have evolved to trick our immune system, and viruses also. So this particular virus, this vaccinia virus, will produce a protein which stops the complement system working, for example. Some bacteria can grab molecules out of the blood, which makes them invisible to the complement system, so they don't get noticed as well. So although this is a really good system, because it's an ongoing sort of arms race, evolutionary arms race, we call it, the bacteria and the viruses are trying to evade our immune system. Our immune system is trying to adapt to catch them all. There's an arms race going on. So now and again, something gets through that can trick our immune system. HIV is a really good example of that. Damages the immune cells, for example. So it's only a really small part of our immune system. There's loads of other things involved. We just had a look at phagocytosis, for example. So it's loads and loads of parts all working together, and this is just one small part of it. Obviously, you'll be able to watch this video with the commentary on, because I've given you the slides for this. So if you want to do that, you can. So next part of this non-specific defense then is the inflammatory response, which looks pretty painful and nasty there. What's happening is our infected or injured cells release histamine and they release prostaglandins. What these do is they promote dilation, so expansion of local blood vessels, so they open up, and that allows more blood to swell in, and it also increases the permeability of the capillaries, so more fluid can swell into the tissues as well. And what this hopes to do is contain the damage or the infection to a specific area. It also can be really painful. Then we have the last non-specific response, which is to raise the temperature. Macrophages that find invading microbes will release a chemical that's carried to the brain, and this causes the hypothalamus to raise the temperature. And what this does is it stimulates phagocytosis, so it helps to stimulate the engulfing of those pathogens. So that's our non-specific response after so first line of response is those barriers, the skin, those mucous membranes, everything that we try to use to keep everything out. Then if they get breached, we have our non-specific response. So just try and kill it in any way we can, the complement system, the phagocytosis, all of those things. And then eventually the last thing that happens is we've got specific immunity, or adapted immunity or acquired immunity. It takes a few days to start. So this is how infections take hold before you start to feel better. You might have noticed like when you get ill, you can feel quite poorly for quite a few days if you've picked up a pathogen before your body starts to fight it off properly. That's because your you've got your non-specific defenses doing everything they can, but they're non-specific and they can get overwhelmed. You to get a really quick, good response, you need a very specific immune response. And that's why vaccines are really good, because you don't have to wait a few days to start. If you've been vaccinated, this just kicks in straight away. Or if you've had an infection previously, it's the same thing. I mean, obviously that's not ideal because you'll have been sick the first time round, but you'll still have immunity. So if you come across the same pathogen again, rather than having to wait a few days for your specific immunity to kick in, it just kicks in straight away. So specific immunity, we have antigens on our cells, as we know, and the molecules on the bacteria are different from the molecules on the surface of vertebrate cells. Obviously, we're vertebrate animals. They're different to human cells. And we recognize these antigens as non-self, and the immune system produces antibodies against these. So as we saw earlier, there are thousands of different white blood cells that can produce different types of antibodies. Some of them can produce different types of antibodies. And these are proteins that have a very specific shape. They're often drawn as little Y because the molecule looks a bit like a Y shape. And these antigens will fit to specific, these Y-shaped molecules will fit to specific antigens, which are the structures that we find on the surface of bacteria or viruses or whatever, anything that's non-self. So what they do is they label up the pathogen. Essentially, if there's an antibody on it, it's acting as a label, and that will cause them to clump together. And it labels it up to get destroyed. It identifies it to the destruction cells of the immune system. If a pathogen is recognized by a lymphocyte, so a cell producing these antibodies, what it will do is divide rapidly and make loads of copies of itself to help it destroy these invading pathogens. So the antibodies are proteins. They're very specific and they match certain antigens. And what they do is they allow the body to recognize that antigen again if it comes across it in the future. So the production of antibodies in the initial infection is called the primary immune response. And if it's during the second infection, it's called the secondary immune response. So we talked about antigen presentation before. We said the special cells that present or show the antigens from the microbe to the T cells. And then the T cells start specific immunity. So let's have a look at that again. So invading pathogen, the macrophage breaks down the pathogen, takes the sort of structures off the surface of the pathogen and shows it to the T lymphocyte. What can happen then is we can activate a killer cell that activates macrophages, which can phagocytose and things like that. But also we can get activation of the B lymphocyte, which produces specific antigens. Whoops, sorry guys. So activation of the B lymphocyte, which produces antibodies against the specific antigen. Okay, and these will stick to the surface. They help to identify the invading pathogens for destruction. They help to flag them up and they help to clump them together. So that speeds up the ingestion and destruction. But also, some of these B lymphocytes that have produced these antibodies can go off and become memory cells. And these can later on, if you come across this infection again, just quickly come out of up to this, the site of infection, whatever, notice this infection and trigger an immune response really, really quickly. So there are tens of millions of different types of T cells that specializes in recognizing a different antigen. And some of these B cells mature in the bone marrow, the ones that I was just showing you. They're specialized, they recognize specific antigens, and they divide very, very rapidly on recognizing an antigen and produce antibodies. They stick to the antigens. You can see them coating the surface of these cells here, marking them for destruction. And then if we get a secondary infection, they'll remember the previous infection and activate an immune response. This was discovered by Jenner, scientists working in 1796, and some other scientists who were working on, well, I suppose he wasn't really a scientist. Yeah. Well, anyway, he was working on cowpox and smallpox, and then eventually that work was carried on by Pasteur, who was working on something called fowl cholera. So let me show you a little video. I'll talk you through it because I know you can't hear. So smallpox used to be really nasty and leave people with horrible scars. It used to kill a lot of people. It was, very dangerous disease. Used to cause horrible scarring. So this guy noticed that it was really similar to the disease that his cows had called cowpox. Cowpox was actually transferred to people who used to work with cows. Um, they used to catch it off the cows, which obviously wasn't very nice. But he noticed that loads of people who worked with the cows who'd caught cowpox and survived it, obviously, because it wasn't that deadly, that none of them got cowpox, none of them died from smallpox. So in the smallpox outbreak, he infected his whole family with cowpox. So he's pretty confident it was going to work. He infected them all with cowpox and they survived. So then this guy, Jenner, wanted to prove this. So he was going to do some actual experiments. So what he did was he took some a cowpox blister from an infected person, cut the arm of a young boy who I think was his gardener, if I remember rightly, just some like poor kid in the village, and infected him with cowpox, so gave him the infection. So he developed cowpox, and then not only, which is extremely ethically dubious, but they didn't worry about ethics boards and things like that in those days. So not only has he infected this poor kid with cowpox, luckily kid survived, he then goes and infects him with smallpox, which is well known to be extremely deadly, but it didn't matter because he was just some poor kid. He didn't actually care. Just infected him with this lethal disease and to see what happened. And he was protected, thankfully, from his cowpox vaccination. And that's a nice little fact for you there. The word vaccination comes from the Latin word for cow, which was thought up by Edward Jenner, who thought nothing of infecting small, poor children with um, deadly diseases. Smallpox has been pretty much eradicated now as well, as a result of this knowledge. There was a mass global vaccination campaign of smallpox, which is, I think it's pretty much gone. There's a few small pockets of it, I think, in Afghanistan and Pakistan, maybe, but it's pretty much gone globally. Yeah, 1980 it was announced it was eradicated, but there are actually a few small. Um, pockets of it here and there around the world. But pretty nasty experiment on some poor kid who got infected first with cowpox and then with deadly smallpox. It must have been pretty scary for him if he'd have known even what was going on. Right, I've just got to get out of this now. So what the vaccine does is it contains either weakened or dead pathogens. So in that example there, Jenna, by infecting and the farmer by infecting everyone with the cowpox, they were infecting them with a weaker virus, but it was a very similar virus. It was very closely related. So luckily for those guys, the immune system, once it had recognized cowpox, it also recognized smallpox. So we can do that by giving bits of the virus in the vaccine, not the whole virus, just bits of it, the surface protein. So this is a bacterium here, and you can see these antibodies are recognizing these square-shaped antigens on the surface. So it is possible to, rather than give the whole pathogen, you could just give the square-shaped structures. We've actually developing mRNA vaccines now, which are the instructions to make the proteins on the surface, which is really good. That's what was going on with COVID. So rather than even giving the proteins that are the antigens, we can just give the body the instructions to make those proteins, and then it will recognize them. So that's really good. So vaccines don't cause disease. They've either got dead pathogens, so they've been killed in the case of bacteria, or weakened, or slightly related, or just bits of the pathogen. But anyway, whatever you have, they're unable to cause disease, but they have the same antigens. When they're introduced to the body, the white blood cells will then make antibodies in the normal way. The B cells will make memory cells in the normal way. And then those cells will be there for long-term immunity. And if you come across that infection again, those B cells can jump into action and quickly respond to the infection rather than having that lag period of sort of 6 days before the specific immune response jumps in. So what happens when you get a vaccine is you have a specific immune response immediately rather than having that 5-day lag, during which time you can get really sick. So some of these vaccines will remain in the blood for a very long time. Depends what the pathogen is. Some of them, like flu will mutate. So the vaccine isn't necessarily that useful because each year there's a new strain. We had that with COVID, SARS-CoV-2 virus as well. That was mutating very rapidly. So after a while, the vaccine that we were all sort of kept going to get wasn't really that particularly effective. But generally, you'll have this very specific immune response happening very quick if you've had a vaccination. So it's really effective. What's good about this is the specific immune response gets activated quickly. The virus or bacteria can't mutate and get a hold. So it's much easier to clear out. So it can just be sort of removed by the phagocytosis much easier. There's less tissue damage, there's less harm, there's less big immune response that can cause damage as well, because the immune response is it's not a sort of gentle process. Often when you get sick, it's the immune response which is making you feel ill, and causes a lot of the damage of the illness as well. Because it's an all or nothing approach. It doesn't do things gently. It just goes right, throw everything at it. And if we kill a load of human cells on the way, so be it. So the less of a violent immune response you have, the better, really. Okay. So what happens then? The person has active immunity to the disease because they've got antibodies which they've made and memory cells that protect against it. You can develop it by recovering from an infection or by being vaccinated. And another type of immunity you can have is passive immunity. And that's when you're given antibodies that have been made by another organism, which might sound a bit weird, but that's what mothers do to children when they breastfeed them to their babies. So babies get passive immunity by receiving antibodies from their mother through breastfeeding. So there's two types of immunity, the active and passive immunity. Okay, we'll leave it there because we're at the end of time, and I will pick it up on this slide next session. If you've got any questions, you can let me know. But otherwise, I will see you on Thursday. Bye, guys. Bye-bye.